Welcome to Hidden Layers, where we explore the people and the tech behind artificial intelligence. I'm your host, Ron Green, and I'm very happy today to be joined by Daryl Barth. We're going to discuss her work on protein engineering and how she's leveraging recent advances in artificial intelligence to significantly speed up the discovery of novel enzymes with the potential to break down plastics, all with an eye towards addressing our global pollution crisis. Daryl's a doctoral candidate in cell and molecular biology at the University of Texas at Austin, with a focus on proteins and protein design. She received her BS in material science and engineering from the University of California, Berkeley, where she first became infatuated with natural materials, sparking a transition into the field of biology. All right, welcome to the show, Daryl.
Thank you, Ron. I'm really excited to be here.
I'm really excited about this too, because I really don't know that much about biochemistry, let alone protein folding and enzymes. But before we jump into all that, let's talk a little bit about how did you get interested in this field? How did you get interested in protein engineering?
Definitely. It was a bit of a long road. So as you said, I studied material science for my undergrad, and I got really interested later on in natural materials because there's a material property called toughness, which is essentially like how much energy a material can absorb. And that is really hard for us to engineer, but nature is really good at it because you need to build in gradients. You have like a soft material on the inside and a hard material on the exterior, and that really allows you to be able to be tough. So things like your teeth are really tough, or I think like one of the toughest materials in the world is like a limpet. Something that, exists on the side of a rock.
Oh, okay.
Yeah.
Okay. Really quickly, because I've never heard this before, is it important that the inside is soft relative to the outside for toughness?
Not necessarily. Okay. It's just the kind of interplay of that gradient. Okay. So it's like if you have something that's really hard and you hit it, it could still be brittle. Like if you think of a ceramic, it's like really hard to the touch, but you can break it really easily.
Okay.
Whereas something more like a metal, it has a bit more of an interplay between the like hardness and flexibility, and so it can absorb a little bit more energy.
Okay, gotcha.
And so essentially, natural materials are way better at capitalizing on those properties. And so I got super interested in natural materials, particularly because they're made at very ambient conditions. Like we spend so much energy and effort on making materials, especially for construction. Like refining steel takes up like, I think at least 10% of the amount of energy that the US produces a year just to refine steel.
That's incredible.
Yeah, and that's not even to mention mining it. Yeah, which is crazy. Whereas you have a spider, and spider silk is close to the same tensile strength as steel, but it's made, in a spider
right.
at 70 degrees, And so I was like, oh, this is amazing. I really need to get into biology. So I spent some time trying to transition after I graduated. So I worked in like a tissue engineering lab, and then I did some more genetics, kind of like cellular reprogramming type stuff, and then happened to land in a lab at UT that focused more on proteins and proteomics.
Okay.
And that's been a really, really cool place to be because proteins are the sort of cellular machines that, make everything happen.
Okay.
Yeah.
Perfect. That was going to be my next question is to kind of tee up the rest of this conversation around protein engineering and protein folding. Can you describe what are proteins and why are they so important to biological life?
Definitely. Yeah. So proteins, I feel like when people first interact with proteins, especially if you work out or something, you think of your big
absolutely.
tub. Yeah, exactly. So proteins fundamentally are molecular machines. So they make most processes in the cell happen.
Okay.
For example, so like if you're trying to transcribe DNA, there's a protein that does that. An enzyme specifically. If you are needing to build muscle, right, you need a protein. So they essentially without proteins, nothing would happen really. Okay. Yeah. So your cells are mostly made up of protein and lipids. So proteins, fats, and then we have a little bit of DNA and RNA.
Okay.
Yeah.
Okay. So it's basically one of the core biological building blocks of life.
Exactly.
Okay.
Yeah.
So I understand that proteins are made from a string of amino acids. Is that correct? Okay. Can you walk us through the process of what what does it mean to take a string of amino acids and produce a protein? And what is protein folding and why is it so complicated to understand?
Exactly. Yeah. So let's see. There's 4 different levels of protein structure.
Okay.
So on the first level, it's literally your string of amino acids. And there's 20 amino acids. And again, if you're in like the workout culture, you've probably heard of like lysine, arginine, those types of things. So those are some of the amino acids. And like the first level of protein structure is what is your amino acid sequence? And then the second level is each of the amino acids has a different property, or kind of groups of properties. So some of them have hydrophobic residues, so they don't like water. They tend to be more oily.
Okay.
Some of them have polar residues, and then some are actually charged. They have a positive or a negative charge. And then others like to form more of the backbone of the protein.
Okay.
And depending on these kind of like molecular interplays, you get different folds that happen. So like first level, you have the amino acid. Second level, you have some structure starting to come from the interplays of these side chains of the amino acid. So you'll have the two, that people mostly talk about are alpha helices and beta sheets. So you have the protein starting to like fold up into a spiral or kind of like laying on top of each other in this beta sheet.
Okay.
And then in some cases, you'll also have like disordered regions too that don't necessarily like fold up into a specific shape. So that's kind of the second level. So you have your amino acid, and then it's folding up into alpha helixy, beta sheet. And then after that, you get kind of like they're called a ternary structure. So you'll have this like alpha helix, beta sheet, and then that folds up with another one. So that becomes like one protein domain. And then to actually form a bigger protein, you might have 2 that come together.
Okay.
And then finally, the fourth part of the structure is even more. So like this is made up of 2 domains. And then this one has like 3 domains, and they come together to form a big molecular machine.
Okay. Can those different parts of the structure, can they all be produced from a single amino acid sequence?
The very like one of the domains is a single amino acid sequence that'll fold up together. Okay. And then, I mean, to answer your question, yes, it gets a little weird. There's some amino acid sequences that'll like auto-cleave into 2 parts and fold up and come together. Right. And you're just like, what are you doing?
Okay, and so the complexity, to the best of my understanding, the complexity of predicting how some sequence of amino acids, which we can think of, whether 20 amino acids?
Yes.
Yeah. So there are 20 amino acids, and they can be ordered, arbitrarily. They're going to be converted into a protein, but that protein structure, based upon whether it's hydrophobic or other properties, will react not only to its environment, but to itself. And so the angles of the molecular interactions is just very complex to predict.
Very complex, yeah. Like we have trouble modeling, small molecules, and this would be considered a macro molecule for sure. So you have all of these like small interactions, compounding on top of each other.
Okay. So even small molecules are difficult to predict, and proteins are definitely in the large molecule camp. Yeah. Okay. So before we kind of segue into how some really recent advances in AI are affecting your world, let's talk about how protein engineering and protein folding predictions were kind of done until recently. I know about there were like X-ray lithography and things like that. Yep. Was it lithography or
crystallography?
Crystallography.
Yeah.
Thank you.
Yeah, you're good. X-ray crystallography, and then a little bit later on, it was cryo electron microscopy.
Okay. What can you describe that?
Yeah. So I don't know if you've seen electron microscopy images. It essentially uses an electron as the wavelength to view whatever structure you're looking at. Okay. And so cryo is freezing. So it's like cryogenic EM. And so electron microscopy used to be used, I guess it still is used for like inert materials for the most part. So if you wanted to look at this piece of wood, you could. After a while, the electron would like burn the surface of it, so it'd be a little hard to see it. But in terms of cryo-EM, the ability to cryogenically freeze something enabled us to look at biological materials, because then you could like preserve the shape and actually view it under the electron microscope. Okay. So X-ray crystallography, that is like diffraction of X-ray beams, and you can essentially figure out like the angles that, like essentially if something hits part of the protein and then bounces off at a certain angle, then you can start to build up kind of where each atom is in space.
Oh, you can infer the positions or the structure of the protein by the deflections of those X-rays?
Yes.
Okay.
So that's X-ray crystallography. And then cryo-EM is literally like a picture. But since you're using an electron, you can get to the resolution of like the wavelength of the electron. So you can actually start to see really, really small objects when you use electron microscopy.
So these are traditional methodologies, but my understanding is that it's fairly slow, right? I mean, somebody might spend years trying to understand how a single protein is folded. Is that correct?
Yes.
Back in the day?
Yeah, particularly earlier on. And a lot of that is actually crystallizing the protein is the hardest part. So particularly for like X-ray crystallography, you need to actually have a crystal to be able, because you don't need, if things are moving around as you're trying to, get beams off of them, then there's no way to back calculate where the thing is.
I definitely didn't understand. I didn't understand that. So when you're going to crystallise the protein, what does that mean?
So I've never actually done this before, but essentially it's like freezing it. So you've made the sugar crystal thing as a kid, like rock candy, that kind of thing. I think it's a pretty similar process to that. You just have to like very slowly go through lots of different steps to try and freeze the protein without denaturing it. Because a lot of proteins are quite sensitive. They're like heat sensitive, they're UV sensitive. So there's all these different things that can make it unfold. So denaturing a protein is when it is unfolded. So it's active in a particular position. Like particularly for enzymes, the shape of the enzyme as well as its like chemical nature is important for its function. And so if you don't have that shape, then the protein won't be doing what it's supposed to be doing. And
so the shape, so this kind of closes the loop and helps a bunch. No, this is fantastic because the shape of the protein determines its behavior, its function, how it interacts. So being able to predict what that shape would be will allow you to do things like, for example, within your work, to predict enzymes that might be able to break down plastics.
Exactly.
Okay, I would love to transition. Let's talk about some of the big breakthroughs. I'm familiar with AlphaFold, but for our listeners, maybe just real briefly describe how are you using it on a daily basis to help in your work?
Totally. Yeah. So how am I using it? So a lot of it is the trying to see the structure in the function. So I guess in research before literally like 3 years ago, which is kind of crazy.
It's crazy.
Yeah. If you actually wanted to see your protein, it would be arduous, and so you would only want to actually look at the protein if you knew this was something like useful that you wanted to characterize further.
You mean take the time to try to really study that protein?
Yes. Yeah. And there were, so before AlphaFold, there also were other methodologies. We talked, I don't know if we should talk about the...
No, absolutely. Let's talk about it. So I've got in my notes here, you're talking about the critical assessment of structured prediction contest. Okay, yes, which has been going on for several decades now.
Yes, several decades. So this whole field was really launched, I think, in the late 50s by a scientist named Christian Anfinsen, and they call it Anfinsen's dogma, which is very fun. But he was pretty convinced that all you needed was the amino acid sequence to determine what the protein structure would look like. And so he actually won the Nobel Prize with another scientist in the 70s for his work on this. And so he kind of launched the field, and people from him were like, oh, I guess maybe you just need the amino acid. And so like the amino acid sequence. And so people started really digging into this. And to kind of converge the field on it, in 1994, they started CASP, which is the competition that you mentioned. And so every 2 years, it was like the protein structure Olympics. A bunch of scientists around the world would get together and try out their models on a new set of proteins where like they had been crystallized. So the people, running the competition knew what the structures were, but everyone testing their models didn't know.
Didn't know.
Yeah,
okay.
didn't know. And so they were trying to see how well they did. And the first approaches were all, physical models. So like we talked about the amino acids earlier, all of the charged, hydrophobic, et cetera, residues, how do you model them in such a way with the physical knowledge that we know to try and get the right structure from all these different interactions?
Okay.
So that's how that started. And it had, kind of like different jumps back and forth. There was some progress, and then there were plateaus and progress and plateaus. But really, it wasn't until AlphaFold came out around the very first iteration was 2018, but the big deal was after the transformer was invented.
Right.
And in 2020, AlphaFold just blew everyone out of the water.
I remember the news. It was really amazing because even though I really don't know much about this field, I do know that protein folding is considered like one of the grand challenges within biology.
Yes.
And AlphaFold really, really kind of blew away, I believe, all previous models in its ability to predict folding. Is that correct?
It did, yeah.
Okay. And then 2 years later, I believe AlphaFold 2 was released.
Yes.
And my understanding is that was, again, another sort of dramatic improvement in prediction. Is that right?
It was, yeah. It was like completely dramatic. And I think the 2018 one was like some progress, but not as dramatic. And then 2020, some people would consider protein folding to be solved, or at least very much on the way to being solved, like the first 80%.
So I remember reading that too, that some people were arguing that, okay, this really difficult problem that's been around for decades is solved. And then some people were a little bit more skeptical since AlphaFold 2, and now I guess the competition will be held again this year.
I'm actually not sure.
But my guess would be there's probably general consensus that this new approach is, if it hasn't at least solved the problem, we're well on our way. Is that a fair assessment?
That's definitely what it feels like. Like the paradigm shift is there.
Okay. All right. So I'd love to, all right. So going from using X-rays and electron microscopes and things like that to now just interacting with a software tool.
Yes.
Can you take us through that process? So you have a sequence of amino acids, which in this instance, I think is literally, you're just saying it's a sequence of letters. And each letter is one of 20 letters that represents that amino acid.
Exactly.
So I don't know if it's A, B, C, D or something like that, but you take that sequence and then what?
Mm-hmm. Yeah. So it's to the point. I think they have an API. There's a few different ways that you can do it. A lot of people have set up collab notebooks. If you've heard of those.
Oh, absolutely.
Yeah. So there's a collab notebook online that you can go to, and you can copy and paste your amino acid sequence into it, and then click through the cells, and you'll have a folded protein.
That's just unbelievable.
Yeah.
How long does it take? Does it matter the length of the amino acid sequence, or is it always the same amount of time?
Definitely matters the length of the amino acid sequence.
Okay.
When it first came out, it was really slowing down, I think, after about 1,500 residues. So, or over at like 1,000 to 1,500.
Wow, that's a lot longer than I thought you were going to say.
Yeah.
That's already pretty impressive.
Yeah. So a really small, or, kind of in the work that I do, a pretty small protein is about 200 amino acids. And then median size, maybe between like 800 to 1,000 for monomers. So we're talking about just like one amino acid sequence to that initial domain that we're talking about. Right. Yeah. They can get huge though. I've seen some like protein complexes people have folded where they're like 20,000 or even more residues, like all put together. So there's some really incredible work that people have done out there, even just with like playing with all the different parameters and folding multiple proteins together and stuff. So that's really remarkable. If you want to go just like look at pretty pictures of proteins, that's definitely possible. But you were asking.
Yeah. On your day to day, I mean, because it sounds like, it's kind of, to me, it's actually one of the realizations of the promise of AI that we've been talking about for decades, right?
Yes.
This idea that AI systems wouldn't just be intelligent and help you, with your day-to-day work. It could actually advance different fields of science.
Yes.
And it sounds like biology and protein folding, your field in particular, it's really making an impact.
Huge.
And so on a day-to-day basis, within your lab, are you all exploring vastly more sequences than you would even imagined like 5 years before?
Absolutely.
And So let's talk about that. And specifically, let's segue into enzymes and plastic decomposition and your work there, because unbelievably important. Let's just talk about how you're leveraging it and how it's helping you make progress on that problem.
For sure. Yeah. So to kind of contrast, I think, well, actually, in my own personal experience, kind of as I've come into this, AlphaFold has been a tool, which is kind of wild. But even as I've been working on it, it's become more and more available and like possible to work with it, I guess. And a lot of other softwares have started piggybacking off of it. So I think the most compelling advance to me is there's a kind of an old school algorithm called BLAST, and it essentially allows you to compare amino acid structures, or like amino acids to amino acids. So you have your sequence, and then you can look through huge databases of all known proteins and see like what other species have the similar protein. And kind of like line that up.
Find the most similar match.
Exactly. And so now that we're able to fold, X amount of proteins at a time, you can now do that with structures, which is like kind of mind blowing and really cool. And so it allows you to kind of go, if you're trying to find similar proteins in the kind of mixed bag of whatever evolution has created in terms of proteins, you can now use sort of two different approaches. You can use a structural search approach, or you can use a kind of sequence search approach, which is neat. So it's sort of like doubled your ability to search. That's important for me because there's a few known enzymes that have been shown to be effective at degrading plastic. And so one of the like research branches that I'm going down is using those proteins that are known and trying to find other iterations of them in areas where you'd expect proteins with the kind of qualities that we'd want. So one thing is when you're trying to break down plastic, typically, if you can raise the plastic to a higher temperature, your enzyme will, like, the plastic will loosen to some amount. There's this thing called a glass transition temperature that it goes through. And that means that the molecules in the plastic have more freedom to sort of move around. And that, in a way, like, increases the surface area and the ability for the enzyme to, like, get there and actually eat at the bonds.
Oh, that makes complete sense.
Yeah. And so, but to be able to do that, you need an enzyme that can handle that heat. And so that's like typically around 50 to 70 degrees Celsius tends to be the sweet spot there. But a lot of enzymes, operate in your body, right? Or more in ambient conditions. And so when they get to 70 degrees, they start denaturing, and that doesn't work out. And so what we'll do is we'll go to data sets that have been grabbed from kind of extremophilic type places. So deep under the ocean with hydrothermal vents where there's microbes that can exist at like 100 degrees Celsius. And we'll get the metagenomic data from that. So all the DNA sequences, predict the proteins that are in there, and then search through those protein mounds for enzymes that look like enzymes that we know that work on plastic, but
incredible.
That might be more thermostable or of other qualities.
All right, let me make sure I understand, because that just sounds incredible. Yeah. So you're basically using the search technique BLAST to go look at collections of DNA sequences from these organisms at the bottom of the ocean, but that are near these these thermal vents. So they have incredibly high temperatures because we know they obviously can survive there.
Yeah.
And so then you're using that to identify potential sequences. And I do have a question about how you go from DNA to amino acids in here a second, but you're using that to identify potential candidates from amino acid sequences that would operate at high temperatures to break down plastics.
Yes. Yeah. So that's one way of doing it. Okay. Yeah. And so in the past, that approach has been available, I think, the metagenomic data is more of a newer phenomenon. So being able to have huge pools to look through is newer. Probably like within the last 10, 15 years, that field has started to really pick up. But in terms of being able to do that blast search, that's been around for, like 20 plus years at this point. So the really cool part of that is like, then I can find with BLAST potential structures that work, and then I can fold them. And I can then compare those folds to the known own enzymes and be like, do they work? And so I can, basically narrow down from like 500 potential enzymes to maybe like 20 that might be the ones that are working, before I spend a bunch of money like actually expressing them and cloning them and seeing if they work.
Exactly.
Like in carbon. Yeah.
That makes perfect sense. How do you go from a DNA sequence of some organism at the bottom of the ocean to the amino acid sequence that you may want to explore as an enzyme
Definitely.
candidate?
Yeah, so that's translation is like the kind of name, which is actually hilarious now that I think about that. That never really occurred to me. That's hilarious. Translation.
Exactly.
Yeah, so there's three DNA, so 3 nucleotides that code to an amino acid. Okay. So that's like a language. There's tables where you can go 3 nucleotides to 1 amino acid.
Each codon transforms into an amino acid.
Exactly. Yeah. And so you're like searching through this genome that you have. There's like a start codon and a stop codon. You see the start codon, and then you go from there, and every 3 nucleotides becomes an amino acid.
So you don't have to go through through DNA to RNA to amino acids. You can go directly from the DNA. Okay.
Yeah.
That was one part that I wasn't sure if you were going through the full transcription process.
Oh, yeah.
Okay.
Yeah.
So, all right. So I'm really fascinated about this next part. So you have some amino acid candidate. How do you reconcile the theory, the prediction of what, let's say, AlphaFold says it will do with the reality of what it does? How do you confirm that? How do you test it?
Yeah. So a lot of that is you have to lay it into carbon, as people say. Yeah, honestly, that's probably the next frontier, is figuring out actual function. So you can how people try and do it is sequence similarity, structural similarity. How close is it to a thing that we have tested in real life? Is kind of the basic. Okay. So when you're trying to get past that, that's when you actually have to go into the wet lab and make the thing that you want to see what it does, and then test it and see what it does.
And so you actually go and make it, and then do you just expose it to plastics at high temperatures and then see how it reacts?
Yeah, there's a couple of ways of doing it, but yeah, that's basically what it is. Like you'll cut hole punches of the plastic and put it in little Eppendorf tubes and keep them at different temperatures, and take time points over days. And then there's this thing called an HPLC that you run it through, and it's called high performance liquid chromatography. And there's a bunch of different ways of doing it, but you can basically figure out whether you're seeing the breakdown products of the plastic in solution. Okay. And that'll give you an idea of like how quickly your enzyme is acting, or whether it's acting at all. Okay. So
that is just amazing. Are there any, like, where are we in the process? Are there any strong candidates right now that are showing promise?
Yeah, definitely. So and this field has been going on for a decent amount of time. I think like early 2010s, and actually even earlier, people were interested in enzymes that could break down plastic, but not as intensely as they are now. I feel like a lot of papers that you see nowadays, they're more industry type focused.
Is that mostly because there's increasing confidence that this can be done?
I think that, and then also demand for it. I feel like before the 2000s, the plastic problem, it was kind of like global warming. People were like, whereas nowadays it's really gathering steam, and there's funding for it, which is a big part of it as well. So it depends on the type of plastic you're talking about. So I think there's something crazy like over 100 million tons of plastic produced each year, which is like kind of unfathomable. Really, I can't imagine 100 million
it really
tons.
is unbelievable.
Yeah, so there's that much produced, and then of the plastic that's being produced, there's about 6 types of plastic that make up the majority of it. By far the most is a plastic called PET, which is polyethylene terephthalate, and that's what you see in water bottles for the most part. And then after PET, you have there's like polystyrene, styrofoam, polypropylene, which is a bit harder plastic, polyurethane, polyethylene, and then nylon are like the full 6. And in terms of PET, the bonds that kind of hold together the polymer there are a little bit easier to attack than the other types of plastic.
Okay.
They're more similar to, bonds that you would find in biology. And so there are more instances of enzymes that can take down PET, which is cool.
Okay.
And so people have really focused on it. The very first one that people found that they actually termed a PETase was in, I think, 2015. There was one before that, which was called a cutinase that actually came out of leaf compost, which is kind of wild. But so in 2015, outside of a water bottle plant in Japan, there was a microbe that was isolated called Ideonella sachianensis, and they found that the microbe was able to live exclusively on PET plastic, and so it was able to extract energy from it and actually metabolize it, which is pretty incredible.
That is incredible.
Mm-hmm. And so from this microbe, they were able to figure out which enzyme was the most responsible for the PET breakdown, and that became a big PETase. So it's called IS PETase after Ideonella sachianensis.
Okay.
So that was kind of the first instance of PETases. And so between the leaf compost cutinase and the one from the outside of the water bottle plant in Japan, you have these two pretty amazing enzymes that people have then done some pretty intense engineering on to try and make them more thermostable. And so literally, I think it was last month, there was a new PETase released called turbo PETase that actually looks like if you put it in a recycling plant, it would be able to accomplish, breaking down PET into its monomers at a scale that you would desire if you were actually trying to do this.
Oh,
properly.
That's incredible. So really close to having something that might be useful at industrial scale?
Definitely. Yeah. And there's actually a company in France who used the leaf compost cutinase one, and they've been working on it for, I think, almost 10 years now. And they have a pilot plant that I think started up a few years ago, and they're scaling it up. So there's actually one instance in the world, but one that can actually degrade PET. Okay. So in terms of that one plastic, PETase, things are going pretty great. There's a lot of hope. For the other plastics, where the road is long.
It's the early days.
But there's possibilities though. There's for each of the plastic types, there are either known organisms that can eat it, or there's been instances in the past where you might be able to work on it. If that makes sense.
Okay. That's unbelievably fascinating.
Yeah. That's why.
Wow, that I am just so impressed, and I'm just so personally so excited that we're at this stage now where people like you who are doing important work are able to leverage AI in a way that I think a lot of people who listen to this are going to be really surprised by just how much it's impacting bleeding edge science.
Oh, it's been amazing.
Okay, this conversation has been unbelievably fascinating. We like to wrap up with a fun little question. Mm-hmm. If you could have AI automate anything in your life, what would you go for?
Oh, anything in my life.
Yeah, anything.
Oh, not just with work. Oh, interesting. Okay.
Yeah.
Oh, you got it.
It can be work.
Oh, wow. Anything in life.
Yeah.
That's pretty cool. I think personally it would be time management. It's a big thing, which I know is coming. Definitely scheduling in and figuring out what to prioritize. Yeah, that would be huge for me. But in terms of the field, and we're kind of coming up upon it. It sort of goes back to the function aspect. We spend a lot of time, the wet lab pipeline is really intense and also expensive to figure out if these enzymes are actually working.
We're actually producing the enzymes.
Exactly.
And testing them. Okay.
Exactly. And so if we had a way of being able to more efficiently or correctly characterize the function of a protein,
mm-hmm.
that would be amazing.
Okay.
And there do seem to be people are trying to get there. People are training classifiers off the end of AlphaFold. So you have
mm-hmm.
your structures come out, and then they'll have a classifier of different similarly functioning proteins, and so you can say does it fit in this pool or this one, that kind of thing. So that's sort of happening. And then there's also like the large language models that are just taking in the amino acid residue, and there's some interesting stuff happening there. But in terms of actually being able to say like, here's structure, here's residue, this is what it does.
Right. Then just to be clear, that next step, there's one thing taking an amino acid sequence and predicting how it will fold.
Mm-hmm.
The next step is, how will that actually behave?
Yes. How does it interact?
How does it interact
yes.
in solving that?
Exactly. Which is huge. And we're approaching that possibility on the small molecule scale. It's actually, I did want to mention this, there's some really exciting stuff coming out recently. There's something that came out, like the code came out for it, I think literally yesterday or a few days ago. It's called RosettaFold All Atom. And so it's essentially like an AlphaFold, but instead of just looking at proteins, it also incorporates small molecules and like ligands and stuff, or atoms. Okay. So then you can throw in, like CRISPR is a ribonucleoprotein, right? So it has RNA in it. I think I said that wrong. Oops. But yeah, so essentially it's like RNA and complex with the protein is how CRISPR functions. And so you could ostensibly fold that whole complex using RosettaFold All Atom. Wow. Which is like crazy.
That's crazy.
Yeah, so that people are like working on it and trying to get there. But that next step, the sort of like, okay, we have the thing, how does it interact with its world? Right. Is the kind of next step. I think that's the frontier we're trying to move.
All right,
next.
we'll make sure to put links in the show notes so that people can go access all the different tools and technologies we talked about today. Yeah. Well, thank you so much, Daryl. This was fascinating. I can't tell you how excited I am about your work and how you're leveraging AI, and for taking the time to chat with us today.
Thank you. Thanks for inviting me. This has been really fun.
Thank you.
Yeah.
Thank you for listening to Hidden Layers. This series is hosted by Kung Fu AI, a management consulting and engineering firm focused exclusively on artificial intelligence. If you have any questions or thoughts about today's episode, or if you know someone we should feature, please visit us at kungfu.ai.